vicepresidente y director de Genómica y Ciencia de Datos de Regeneron Genetics Center, con una conferencia que ha denominado Aventuras en Genética Humana. El doctor Gonçalo Abecasis supervisa la genética estadística y la ciencia de datos cuantitativos, impulsando nuevos descubrimientos genómicos en el Centro de Genética Regeneron. Antes de unirse a Regeneron, fue miembro del Consejo Asesor Científico y director de la Iniciativa de Genómica de la Universidad de Michigan y profesor colegiado de bioestadística Felix Moore. En 2014 recibió el premio Kurt Stern de la Sociedad Estadounidense de Genética Humana por sus contribuciones al análisis y la comprensión de rasgos humanos complejos. Y en 2013 obtuvo el premio Overton de la Sociedad Internacional de Biología Computacional por las herramientas y métodos que desarrolló para el análisis conjunto de Big Data. El doctor Abecasis obtuvo su licenciatura en genética en la Universidad de Leeds y su doctorado en genética humana en la Universidad de Oxford. Bienvenido, doctor Gonçalo. Muchas gracias.
Thank you very much. Pleasure to be here today in the middle of such a great roster of speakers. So I wanted to talk to you today a little bit about human genetics, what our goals are in human genetics to improve medicine, give you some examples, and also talk a little bit about the challenges where innovation is required, and a little bit of where you need luck sometimes. So I wanted to put up these two quotes. You just heard in the previous talk about how the human genome has now been sequenced for almost 20 years. At least the 99% of it that was easier to sequence has been sequenced for almost 20 years. And so we now have a complete catalog of all the parts, all the genes that make up each one of us. But it turns out that if you wanted to know what the function of each of those genes is, if you wanted to know what would happen if one of those genes broke, most of the time we don't know. And I think that quote captures it well, that there's a lot of genes, there's over 20,000 genes where we'd like to understand function, and what we know is probably just a little bit. So we hope to shed a little bit of light and understand what else is going on. I think the other thing that I wanted to make a point from this quote is that, understanding the health of individuals, why some people get sick, why some people don't, can tell us a little bit about the health of a population. Maybe we don't need to rely on death, as in the quote from Octavio Paz, but we do rely on understanding sickness and observing those individuals and using their misfortune to help everyone else. So the goal of human genetics, there's many different goals many different things you could accomplish with human genetics. You can study the history of populations. You can study human movement from one continent or one region to another. Today, specifically, I'm going to talk about how human genetics can help us understand what happens when we change biology. If I modify a gene, what would be the consequences? And I hope I'll leave you with the idea that this can both help us develop new treatments, develop new medicines, and as you heard in the previous talk, it can also give us clues for who should receive those treatments, how can we best improve the health of a population in an effective manner by matching each treatment to the patients who need it. So as a little thought experiment, I like to start with these kinds of questions. What if we could know what happens when we turn every gene on or off? We imagine that maybe some of those genes would reduce our risk of cancer if they were turned off. We would imagine that maybe some of these genes would change how we think about food. They might improve our metabolic profile. Some of them might keep us hearing or thinking better as we age. And of course, one way to imagine doing this would be to do the experiment. You could take a list of the 20,000 genes and turn each one of them on or off and observe the consequences. Now, when you do those experiments, usually you're working with mice or with model systems that are convenient, and they don't always match what happens in humans. It turns out that nature has done many of these experiments for us. Each one of us in this room has one gene or a few genes that nature has modified in a very unique way. And so instead of doing the experiment, we can try and observe the experiment of nature. We can study many people, look at their DNA, and look at the consequences. Okay. At the Regeneron Genetics Center, this is exactly our goal, to observe these experiments of nature, to look at the health of many individuals, and see what we can learn. At the moment, we've looked at the DNA of over 2 million individuals. Out of those 2 million, about 500,000 are non-European, so we would consider them to be understudied. They're sometimes from populations like in Mexico, sometimes from populations in Asia or in Africa. But if you think about the balance of human population, there's, out of the 7 billion of us that are out there, probably, less than 500 million are European in origin. But they are, in this case, instead of being 10%, they're 75% of what we've studied. So I think there's a big gap in our knowledge that, hopefully this project will help tackle. We have about 100 collaborations around the world, some studies looking at many diseases, some studies focused on individual diseases. And you heard that, obviously DNA is very important, but it's not all of biology. In the previous talk, we were talking about what is the conformation of chromosomes within cells. We are also interested in expanding beyond sequencing DNA and looking at other kinds of biological characteristics of individuals. And at the moment, we have about 50,000 individuals where we've done deeper genomic characterization. To accomplish these sorts of things, what is required? One of the things that is obviously required is a very effective lab process, both on how you might store your samples, robots that handle the preparation of all the samples that go through the lab, recent sequencing instruments so you can generate a DNA analysis at scale. And one of the things that we decided to do early on was that we would do all the computing on the cloud. So right now, all our computing happens on the Amazon cloud, and that makes it scalable. It makes it easy to keep up with advances. But in parallel, and this is the area where I work, in parallel to thinking how should you improve the lab? What kind of robots should you set up? Where should you place your computers so that they have the power and cooling and the analysis capacity you need? Well, you need to think about what are the analyses that you're going to do, both from the mathematical and statistical side and from the computational side. I wanted here to highlight as an example some of the work that two of my colleagues, Joel Mabacho and Jonathan Marchini, have done in making sure that if you could generate genetic data on 2 million people and have the health information on 2 million people, you could interpret it. So they took mixed model approaches that are kind of current standard approaches for modeling genetic variation while accounting for the background effects of population, the background effects of relationships between individuals, and answering questions about what are the effects of changing a specific piece of DNA. And they took these approaches that are now quite well established, and they re-engineered them. And they re-engineered them in a way that we went from analysis that took something like 1,000 hours to analyze a single trait on 500,000 individuals down to 10 hours. That is a pretty important advance because it means that in a setting where we might measure thousands of outcomes across millions or hundreds of thousands of individuals, that will translate into a savings of hundreds of thousands of dollars, sometimes millions of dollars. And Pablo had this quote about figuring out how you make the impossible possible. Well, part of it is by continually innovating how you do the analysis and think about these problems. Okay, so lots of people have heard about Regeneron and the Genetics Center through the work we've done with UK Biobank, and obviously that's been very exciting. But I wanted to tell you a little bit more about what happens behind the scenes, give you a quick example of how we generate data, some examples of the discoveries and how we imagine applying them. Okay, so what kind of data do we generate? Let me just... Okay, so if you imagine this is the DNA sequence of several individuals, and you imagine that you have, as you go along a chromosome, there's some parts marked here by these boxes, which are the coding regions of DNA. Then you have the intervening DNA that also has variation. Well, let's now focus on a single individual. We start with exome sequencing of every sample. So we try and generate a lot of detailed sequencing reads for every piece of coding DNA. So we know about all the changes that modify a protein that, for example, might remove the function from a protein. And then we layer on top of that genetic data on the intervening regions as well, so that we know not just what's happening in coding regions, but what's happening everywhere else in the genome. And we're running these in the lab in a single automated assay. So the samples will be loaded into a large robot. The robot will prepare all the sample, both for the exome sequencing and for the sequencing of intervening regions. This goes in a sequencer, and you get the results at the end. We typically are measuring about 1.4 million common non-coding variants in addition to all the coding variation in each sample. We are getting quality that we think is comparable to an array, but we have a process that's very scalable, allowing us to do up to those 1 million samples a year. And I won't go into this in detail for now because I want to get to some more interesting results, but that we think beats what's possible with genotyping arrays measured in blue in terms of quality for a range of rare to common variants. Red is the performance of our assay, and that works well in a variety of populations. This approach based on sequencing has been designed that it works well not just in Europeans, but in African ancestry, Native American ancestry, ancestry from different parts of the world. Okay, now when this all works well, what are we hoping to learn? What is the goal? What are we trying to get to? And so let me take you through a couple of examples. So we reported on a study a couple of years ago where we found that for this particular gene, a gene called GPR75, this is a G protein coupled receptor. It's important for signaling in the brain. About 4 people in every 10,000 have a defect in this receptor. It does not work properly. Now, if you look at many, many people, in this case we had looked at about 600,000 individuals, 4 in 10,000, there were 228 individuals where the receptor was broken. And the result among those individuals was that their body weight was about 12 pounds lower, about 5 kilos. And as far as we could tell, there were no other bad health consequences. If you are missing 1 copy of this receptor, you're more or less healthy, but you are lighter. This graph here compares GPR75 with a reduction in BMI of about 2 units with other genes where rare variants have a big impact on obesity. MC4R is a very well-known obesity gene. If you have a defect in MC4R, your body weight is almost 2 BMI units larger. The calcitonin receptor also increases weight. GIPR and GPR151 lower your body weight. We actually followed up at that time this experiment in looking what happened at mice on a high-fat diet. So if you look at mice on high-fat diet over a period of several weeks, you'll see that normal mice, the wild type in the black line, gain weight quite rapidly. If they are missing one copy of GPR75, they gain weight more slowly, despite being on the same high-fat diet and having the same availability of food. And if they're missing both copies of GPR75, they gain weight even more slowly. Also interesting, if you challenge them with glucose, with sugar, they have a better metabolic profile when they have the knockout. And so currently, both at Regeneron and in collaboration with colleagues at AstraZeneca, we're trying to pursue whether it's possible to design medicines that block GPR75 to get these kind of benefits, a better metabolic profile, protection from diabetes, and loss in weight. And we got the idea of doing this among all the possible G-protein coupled receptors by observing these 200 individuals out of 600,000 that had a natural defect in the gene and had all these benefits. Here's another example. So in this case, there's a pathway involving the interleukin-4 and interleukin-13 genes, IL-4 and IL-13, that's sort of essential for allergic immune responses. It's one of the key steps in the pathway is signaling through interleukin-4. It's now well known, because there are very successful, very effective medicines out there that act by blocking IL-4 and IL-13, that this is a very good way to work against severe asthma. If you have a patient that has frequent and severe asthma attacks and you can block IL-4 and IL-13, they're much less likely to end up in hospital. They're much less likely to present with an asthma attack. And so we took genetic variants in the IL-4 and IL-13 regions and tried to group people into 5 bins, depending on exactly what set of variants they had near IL-4 and IL-13. Here's a group that has lower risk of asthma, some groups with intermediate, and the group with the highest risk of asthma, depending on genetic variation in the IL-4 and IL-13 locus. Now, in this case, you already know that IL-4 is an effective drug target. It has benefits. But it's now interesting to say, if we look at these exact same individuals, what else do we know about their health? And so in this case, if you take the same groupings of individuals based on asthma around IL-4 and IL-13, you'll find that the low-risk individuals for asthma, they also have lower risk of atopic dermatitis and eczema, another allergic disease. And if they have the genetic variants that put them at high risk of asthma, they're also at high risk of atopic dermatitis. If they also have lower risk of nasal polyps and higher risk in the high-risk group. The same for eosinophilic esophagitis, kind of inflammation in your digestive tract. The same for prurigo nodularis. It turns out that all these 4 outcomes in green, where you can recapitulate what you see in asthma with these other diseases, are places where you can use the exact same interleukin-4 blocking drugs and get a benefit. So now we started with this drug works well for asthma. We were able to search for other indications. And in these indications that I'm listing here, actually the trials have been done and they've been demonstrated to be successful. So you've identified new places to use an existing medicine effectively. You can also see that, for example, if you look at other types of immune diseases, rheumatoid arthritis, actually there, you'd expect blocking interleukin-4 would not be effective, because when you look at people that have been separated depending on their type of interleukin-4 signaling, they have about the same risk of rheumatoid arthritis. Same for Crohn's disease. So there will be a set of diseases that signal through interleukin-4 where we can recapitulate the asthma association. There's other diseases where it won't be effective, but it's much quicker and safer and more effective to do these experiments first by looking at human genetic data and then doing the clinical trial than to do the clinical trial to test these hypotheses for the first time. Okay. I also want to say that, you need a little bit of luck. If you are methodical about doing research and studying interesting questions, you can end up making very important contributions in areas where you don't expect. And so I want to talk about that with two very brief examples. So in the middle of the COVID pandemic, Regeneron was one of the first companies to develop an effective treatment for COVID. So this is more or less the timeline. Oh, sorry. Can I go back? Okay, sorry. In January 2020, just at the beginning of the pandemic, we started a program to try and develop antibodies for COVID. In this case, we started both by immunizing mice with COVID and by collecting serum from patients that have been infected with COVID in Asia. By March, we had several candidate antibodies that we thought would block and neutralize COVID. By April, we had picked the top candidates, decided to make a cocktail, and then in June began human testing. And by the end of the year, these had been tested, approved, and were available for use. Obviously, the virus mutated, and it turns out that as of now, and since the middle of last year, these antibodies are no longer effective. You'd need a new treatment. But what made it possible to so quickly for us develop these treatments? Why didn't other companies do it? Everybody was trying, everybody was keen to solve this problem. It's a worldwide pandemic. So it turns out at Regeneron, one of the reasons that it was possible to so quickly develop these treatments for COVID is because we had developed the expertise in rapid treatment in trying to tackle Ebola epidemics several years before. And actually, it turns out that my colleagues who were trying to develop Ebola treatments, the first few times they tried to develop a treatment. By the time they had the treatment produced at scale, the Ebola epidemic was done. So there's nowhere to try it. And so they had worked very hard to try and get that interval as short as possible. And they had also worried about problems like, what happens if the virus mutates? How do you keep the treatment effective? And so they had landed on this idea that it's very important to have a cocktail that tries to hit the virus from multiple different angles. And so starting with a disease that's in the middle of Africa, pretty rare, figuring out how can you get the treatment there fast enough for each outbreak that probably affects several hundred people, enabled us to have the technology and the expertise to develop a treatment for COVID that actually affected, tens of millions of people. Probably most of us in the room were impacted in some way. I wanted to give you another example about how, sometimes treatments and cures and good ideas come from unexpected places. Oh, sorry, this is just, I forgot it. I had these couple of slides. So we've also obviously tried to tackle the genetics of COVID in collaboration with many people around the world. One of the early findings was that there's a small group of people that have a genetic variant in a gene called ACE2. This is the receptor for the virus. That actually means they produce less of this receptor. And if you're one of these lucky people, about 1 in 300, your risk of COVID is noticeably smaller. And this is an effect that we can reproduce in people of different ancestries from different studies around the world. And now we have even more data about this. You could imagine that potentially in the future, another way to prevent COVID infections, instead of blocking the virus, might be to block the ACE2 receptor. For those of you who are curious about how the epidemiology and the genetics of different diseases compare, we were curious at the time to see how does the genetics of COVID compare with the genetics of flu. And the short answer is that they're totally different. It's a totally different set of genes that influence whether you have a severe flu event and how you respond to flu versus how you respond to COVID infection. Okay, many years ago, about 2004, 2005, together with some colleagues, we had decided to do a detailed genetic study of a population in this Lanusei Valley in Sardinia. This is a picture of the valley. We, at the time, recruited about 6,000 individuals from a total population of about 10,000 in the Valley to participate in a genetic study. In that study, some of my colleagues were very, very interested in understanding what would regulate different kinds of hemoglobin levels in each of the study participants. And one of the early discoveries, there's a paper from about 2008, showed that people with defects in the BCL11A gene kept making fetal hemoglobin into late life. Fetal hemoglobin is the hemoglobin that most children make when they're very young, and they usually stop making it around age 1. But in a few people, they make fetal hemoglobin throughout their whole life. Now, they were interested in this because they had an hypothesis that if you kept making fetal hemoglobin, you would be protected from the effects of thalassemia, which is a common hemoglobin defect in Sardinia. And actually at the time, we were able to confirm this, that, if we took our genotype that was associated with people who kept making fetal hemoglobin into adulthood, this was much more common in individuals who had minor thalassemia. This is thalassemia with no severe health consequences, versus in those who had major thalassemia, thalassemia with severe consequences needing blood transfusions every week or so. Now, this is a finding from 2008. Many of you have heard, for example, that in 2011, 2012, CRISPR technology enabled editing of genomes, and actually just in the last 2 or 3 years, colleagues at Vertex took this idea, edited genes in patients, and showed that, actually if you edit out BCL11A, you can have a apparently permanent treatment for people who have sickle cell or thalassemia. So we started by studying a population in the middle of nowhere, discovering this interesting genetic finding, and now with technology that did not even exist when we started the study, there are treatments out there. And wouldn't we be so lucky if by studying populations here, we could get some clues for treating major important diseases. Okay, I think the clock tells me that I'm a little bit out of time, and so I would just say that, you need creativity, innovation, data, some good luck, and that I really look forward to all that we are going to learn by doing this fantastic and important study together. So, thank you.
Thank you, Gonçalo, for this fantastic talk, giving us an overview of how the genetic information can be used to solve problems. Thank you. One of the questions I have here is how do you think because you showed us that you already have in Regeneron 2 million people, 500,000 of them are not European. So how oriGen will contribute to the goals of Regeneron?
Okay, so I think there are 2 things that are really important. Okay, there are 2 things that are really important when you think about these studies. One is the health problems of different populations are not all the same. If you study the causes of diabetes and obesity or cancer in the US, some of them will be the same as they are in Mexico. Some of them will be different. There are some diseases that are more common in different parts of the world. So if we want to help people in Mexico deal with diseases that are more common in Mexico than in the US, you need to study them. You're not going to do that by studying only people from the US or from Europe. That's one part. The other part is the set of genetic variations that exists in each part of the world is unique. So for example, when we've done studies of what regulates the risk of diabetes in Asia versus in Europe or the US, you make unique discoveries in each population. There's a gene called PAX4 where you can find variants that really change the risk of diabetes in Asia. It turns out that when you study Europeans, there's no variation in that gene. You could study millions and millions of people. You would never know what happens if I modify PAX4, because it turns out PAX4 does not vary very often in Europeans. So each time you study a different population with different genetics, you have a chance to learn completely new things that you cannot learn in other parts of the world. So I think it's both having more genetic variation helps, and I think if you want to study and help different people, you need to study their problems directly. So.
Yeah, thank you. And that highlights also that the Mexican population is highly mixed, and so it's completely unique in the world. So there are a lot of opportunities to try to find new discoveries, right?
I believe that,
yeah.
100%. So.
Fantastic. I don't know whether there are questions here in the audience. Rocío, por favor.
Thank you. Thank you, Gonçalo, for the presentation. And in cases like you present that the gene modify the disease and the outcomes, in these cases, the pharmaceutical company will try to make some products in order to alleviate the disease. But if we are analyzing different populations, my question is, how is going to deal these companies if some population have specific variants with a specific response, and is going to stratify these medicines because now one company may one drug for the whole world, no? But with these differences, how are going to deal with this difference? Are going to prepare many types of, or what? It's one question.
Okay, so I think it will depend on the situation. You know, so for example, if you think about GPR75, you know, we've seen 200 individuals that have a GPR75 defect. These individuals are very different from different populations, all different kind of things happening with their genome. And the thing that they seem to have in common is they're less heavy, they don't gain weight as easily, and they have a lower risk of diabetes. So we would imagine, you know, obviously it has not been tested yet, that if you designed a drug that blocks GPR75, it could help anyone. It could help, you know, it would be an advantage for almost anyone in the modern world to have GPR75 and be able to manage their weight better. Now, there's other kinds of findings, you know, so you might find that in the case of COVID, that interferon signaling is important for developing COVID in some patients. Now, maybe if you said, you know, this increases risk in some patients, now you have a drug that manipulates interferon signaling, it's only going to help those patients. And I think you're going to have some examples like that. So you're going to have some cases where we can figure out what's going wrong with someone and have a drug that's very specific. And you're going to have other examples where you're going to discover a universal principle and something that can be helpful to anybody based on the genetics. And you're going to take the idea you got from those and benefit everybody else that doesn't have that natural protection. I think, you know, only time will tell whether it'll be more common that you come up with drugs that are specific to a group or a type of individual, or more examples where you find a universal principle that then you apply to many people. So.
And one reflection about the that you say about CRISPR, that we can erase the mutation, is, do you think we are going to change the velocity of evolution? Because if we are knowing variants, and we are erasing the mutation with the time, we are going to change this kind of...
I mean, okay, so this is a more philosophical question. So I'm not the best at philosophical questions, but I would say that, you know, if you think about human history, you know, evolution is always changing. You know, if I lived 10,000 years ago, and I had to hunt for my food and fight with other people to stay alive, I don't know. Maybe I would not have been very evolved for that situation, But now we live in cities, we can buy food in the supermarket, and I can work in a computer. It works great for me, So I think the fact that more people can do well over time, maybe people that now have some challenge that that makes their lives very difficult, that in the future we can treat, and now evolution doesn't need to worry about that. That's okay. But yeah, these things are always changing. What's best now and what's best tomorrow could be different. So.
Hola, Gonçalo. Gonçalo. How do you think about the imputation of the genome or the exome starting with a GSA chip for the studies of clinical exomes, if you have some limitation in the very low frequency variant versus, for instance, the using the imputated genome or exome to the PRS studies or applications?
Sure. So we think that there's a good place for both of them. We think that for coding variation, where you want to know what's happening to every protein coding gene, and as you saw in the GPR75 example, we found 200 people that had a knockout in the gene, and there were maybe about 70 different knockouts that accounted for those 220. That's way too rare for what you could do by imputation. So if you want to look at all coding variants, you want to look at gene knockouts, you really just have to do exome sequencing or whole genome sequencing, or you're going to miss too many interesting things. Now, when you want to look at common variation at regulatory variants, you should use imputation, and we do. And I think that's, you have to find that right balance. But we think, look at the coding regions completely, do the exome sequencing, and then use imputation and common variants to get you the rest of the genome. And that seems to work very well for us. All right. Thank you.
Fantastic. Gracias. Hold on. Just a present from Tech.
All right. Thank you.